The first report of a strange new heart disease began in 1990 among the chaos and carnage of a huge earthquake in Japan. Hospitals were inundated. The heart, as it contracted, showed a shape they had never seen before. The bottom of the heart was almost immobile. Many of them recovered completely, with no sign of cardiac damage. They went from an emergency admission with severe chest pain and acute heart failure to walking out of the hospital disease-free. Most never had a problem again. Welcome to the podcast where we dive into the pages of popular books in the life sciences with the writers who made them happen, from new releases to timeless classics. This week, I'm delighted to welcome a world leader in cardiac research, Sian Harding, Emeritus Professor of Cardiac Pharmacology at Imperial College London's National Heart and Lung Institute. Over her 40-year research career, Sian has investigated how heart muscles function or fail in conditions like heart failure using groundbreaking approaches such as gene therapy and stem cell-based repair. Sian led the UK's first clinical trial in gene therapy to improve heart muscle function and is also an expert on broken heart syndrome, which he discusses in her fascinating book, The Exquisite Machine, The New Science of the Heart, our topic of discussion today. Welcome, Sian.
Thank you very much for having me.
I confess that after reading this book, I have a newfound appreciation for just what an incredible organ and perfection of engineering the heart really is. In 1962, John F. Kennedy famously challenged the scientific community to land a man on the moon by the end of the decade. And you reveal in your book that in 1964, a cardiovascular surgeon persuaded President Lyndon B. Johnson to fund a race to successfully make the first functional, self-contained artificial heart before the moon landing. And here we are, 60 years later, and a reliable, off-the-shelf artificial heart is still out of reach. And perhaps that should come as no surprise, because the heart has been more than 500 million years in the making, a long time for evolution to get things right. Every day, it pumps an astonishing 7,600 liters of blood. And of course, the heart does break down. And it's very likely that either you or someone you know has been touched by heart disease. And that's because globally, heart disease is the greatest cause of premature death. And cardiovascular disease, which includes heart disease and strokes, accounts for 32% of deaths worldwide. And today, we'll be talking about the latest advances in science that are opening up the heart's many mysteries, such as growing heart muscle in the lab using simple skin cells. We'll talk about how and why you really can die from a broken heart, whether it's losing a spouse or watching your team miss a penalty, whichever tragedy hits you harder. We'll talk about the remarkable differences in how the hearts of men and women can respond and learn about the heart's own mini-brain, how the heart not only responds to our emotions, but creates them as well. So, Sian, you start the book by talking about the link between poverty and even how we perceive our rank in the social hierarchy can affect the health of the heart.
Yes, there's a really very clear relationship between your social status and your susceptibility to heart disease. Poverty can be a killer in that respect. And it's often thought that this is a lifestyle thing. And I guess there's some contribution from this as well, that people are smoking more or not exercising enough. But even when you pull out statistically these kind of things. And of course, we can do that now because we've got huge data sets like UK Biobank, where you really follow 500,000 people for many years. And you pull out those things, and there is a sort of residual effect of your place in the social hierarchy. British civil servants, the Whitehall study. That's a classic study. And there's a sort of very hierarchical effect on death, that the higher up the rank you are, the longer you're going to live. And I have to say, I think the clearest indication of this is with mice. You can see that mice in a cage, if you put male mice in a cage particularly, they have a very hierarchical structure. You have an alpha male, and the mice at the bottom of the hierarchy can develop spontaneous atherosclerosis simply from the stress of their place in the hierarchy. So that's a little bit difficult to know what to do about.
And it really does seem to be about rank, not even money necessarily, because in that study you mentioned of British civil servants, the difference between those in lower ranks and higher ranks in terms of the salaries they brought home was fairly minimal. And yet they still found differences between those two groups, the higher ranking positions being healthier and living longer, even into retirement and old age.
And you might think about stress, but typically those higher ranking positions are thought to be high stress positions, and then the people in the lower ranks would be comfortably off. They'd be have good housing. They wouldn't be worried about money, and so it's a sort of narrowest social spectrum you can possibly get, and yet you could still see that in those. I did see a statistic once that this works for pretty much everything, pretty much every kind of profession where you could have that kind of ranking, lawyers or whatever. They said the only exception was university professors. And I think just because I remember how what the health and safety was like when I was starting at work compared to what it is now. So I suspect I just inhaled many noxious chemicals. So probably my lifespan has been cut short.
Well, let's hope not. And of course, social status also is related to where one lives. In your book, you talk about air pollution. Now, premature deaths worldwide due to pollution are estimated to be between 8 and 10 million every year. That's more than war, murder, malnutrition, or road accidents. And we think that at least 50% of these deaths are from cardiovascular disease. And you mentioned a fascinating study in London that kind of illustrates the link.
That was done at my place, Imperial, yes. Oxford Street is the main shopping street. And it did have, at this time, a lot of diesel buses and cabs going up and down it. And at the end of Oxford Street, right at the very end, is Hyde Park, which is a lovely big green park, about the same length as Oxford Street, in fact. And so when you measure the emissions in Oxford Street, they are higher than in Hyde Park, but not by, 10 or 20 fold. Because we're still in London, we're still in the centre of London. But yet people in this study who had been asked to walk down Oxford Street for a couple of hours, just at their own pace, some who had health conditions and some who didn't have heart or lung conditions, some healthy people. And they were all monitored up, and you had monitors from the pollution in the street itself and things like that, and the noise. Of course, noise is another big factor. And they walked down there for a couple of hours, just as they at their own pace. And there was a measurable decrease in their blood vessel function and their lung function during that time. And that lasted for some time, for hours to days afterwards. When you walked in Hyde Park, there was a measurable improvement in those things. And again, that lasted for a couple of hours, perhaps a couple of days afterwards. So even in that really very, very short time, in really quite not anything that's so different, you could measure the effect.
And as you mentioned, the difference in air pollution levels between those two locations was minimal, yet it was enough to create this stark contrast.
Yes, absolutely.
And you mentioned their noise, and interestingly, noise levels in Oxford Street also contributed to poorer blood vessel health.
And that's known for people who live around airports, that there's measurable effects on their health from the noise levels around airports. That disturbance of sleep and things. Although clearly in Oxford Street that wasn't the case. Nobody was falling asleep there. So
Yeah,
even just that noise, yeah.
And I guess shows also how urban design can harm health. Reduced green spaces, busy roads near schools, for example.
There's a lot of work on the geopolitical effects of neighbourhoods across the world, and really showing how these very, very small local changes. London, I don't know, people know, there's some very poor areas next to some very rich areas.
Mhm.
And it really is amazing how much difference that makes.
Well, I'll be buying an air filter after we finish this recording,
And
Charlotte.
noise cancelling earphones as well.
And
Yeah.
noise cancelling earphones too. Now, we're currently seeing a diabetes explosion around the world. Of course, food is also related to social rank and poverty and so on. And more than 2/3 of deaths in diabetics are due to heart disease because the high glucose level damages the blood vessels, which impacts the heart.
Mm.
And in your book, you talk about how what our mothers ate during pregnancy can strongly affect the chances of developing diabetes.
Yes, you very often see very high levels of diabetes and obesity among indigenous populations, particularly populations who've had starvation, periods of starvation in their very recent historical past. Theory is that they turn on the genes for making the most of your food. So you metabolically you're very efficient, but also the behaviors that make you want to eat a lot. You're keen to put on fat. Your body wants to put on fat. The body preserves its weight, as they say, because of your behavioral aspects. So when people are now coming into a high food density environment where they've got very calorific foods, food is very easy to get, then it's extremely difficult for them not to put that on.
Yeah, and it's a good example of how our evolutionary past is harming us more than helping us. Because these changes to the DNA that happen during pregnancy in the child in the womb, that kind of senses the environment of the mother, the lack of food will change the DNA, and this will cause what are called epigenetic marks. And so, yes, as you say, the child will then not only be more likely to put on weight, but actually show behavioral modifications to craving high calorie foods, for example, eating past the point of fullness. And so all of this combines to increase the likelihood of them developing diabetes, and especially in countries with recent histories of food scarcity like India and China.
Yes, that's right. And one thing I think is a theme through the book is that the things that have harmed your heart can come from aspects of your biology which were previously survival mechanisms, but they're just inappropriately activated in a time when they're not needed.
Mm-hmm. Well, we're going to get to an example of that later, actually. And just coincidentally, today a lab at the Champalimaud Foundation published a paper in the journal Science, which reveals a surprising new role for immune cells in regulating blood sugar. It seems we still have more to learn about how we regulate our blood sugar levels and the impact this has on health.
I look forward to reading that. Yeah.
So, scientists studying inherited heart conditions have found that family members with the same genetic mutation can show very different symptoms, from severe disease to none at all. And you talk about how this may be due to a so called second hit, which could be some other factor which then combines with the mutation to bring on the symptoms. And that second factor could be consuming alcohol, it could be going through pregnancy, receiving cancer treatment. And a striking example you cite of these second hits is for mutations in the protein titin.
It's very common simply because it's one of the longest proteins in the body. It's basically in the cardiac muscle cell, each cell contracts when the heart beats to pull in the heart, and then it relaxes again. And titin is the protein that pushes it back into its relaxed state, which is obviously important for filling with blood again. And again, I know this very much because it was studied at Imperial, that my daughter was one of the control group in this, actually. The group who was studying this had found that when you've got a dilated cardiomyopathy, where your heart gets big and baggy, and it's the muscle of the heart that's the problem. They were doing some genetic screens, and they found that about 25% of people who had one of these had a problem with the titin. They had one of these mutations or variants in the titin. But they had a control group, And they found that about 1% of the control group also had similar variants. So, looking at the sort of life course of people with the mutations, that's when they discovered that actually you could be perfectly fine. But when you get something else that stresses the heart, then you've just not got the resilience in the heart, and it can trigger this kind of heart failure.
Yeah, it's quite rare actually that we find a direct link between a single mutation and a disease. It seems that many different genes working together normally is the culprit.
Well, when you think about 1 in 250 people is going to be born with a cardiac mutation that has potential to be harmful, they have to be, by definition, mild in a way. I mean, some, of course, are severe, but those that are passed on and on and on can't be too bad because they wouldn't persevere in the population. But most of the time, and particularly in your evolutionary past, you'd never get to the stage where they were going to be a problem. And it's now we're living a lot longer, many more things are happening to us, that you're seeing the problems there.
Great. And so you talked about the protein titin and a little bit about how it works. So maybe now would be a good opportunity to just give people a brief lay of the land for those who may not know much about the heart structure and function.
And so you've probably seen the picture of the heart, and you've got the bulbous part at the bottom, and that's the bit with a really thick muscle. Those are the ventricles. And there's a left ventricle that pumps the blood around the body. As it goes through the body, oxygen is taken out of it and used for all the things your body needs. And it comes back to the right ventricle, which is slightly weaker ventricle. And from there, it goes through another circuit, the second circuit, through the lungs, and comes back into the left ventricle to go around the body again. So you've got this dual circuit. You've got on the top, you've got little atria which help the blood get through and push into the ventricles. So you've got that circuit. And in the structure of the ventricles, what you have is some pacemaker cells which keep the heart going. If you take the heart out of the body, it'll carry on beating, which is very spooky. Of course, it's what I rely on for my experiments, going to the operating theatre and people who've had heart transplant, we can get the failing heart and we can put it on a rig in the laboratory and put even salt solution around it, and it'll just beat away. And so it's got its own intrinsic rhythm. So pacemaker cells give you the intrinsic rhythm, and then there's the sort of workhorse cells, the cardiomyocytes, which have these muscle fibers in that contract and relax. And they contract a bit like your arm muscle, your voluntary muscle, but they have to relax. They have to have safeguards in so that they relax in a regular way so the blood can come into the heart and the filling can happen correctly.
You mentioned the operating theatre, and there's a passage in your book where you talk about the extraordinary moment when during a heart transplant you basically have a person without a heart who's being kept alive by these machines, and it must be extraordinary to witness.
It is. It is a very disconcerting thing to watch. And of course, the surgeons are also blasé about it, having seen it many, many times and know
Yeah.
it's perfectly fine. Yes, it is for a young scientist going in there. It's weird.
Okay, so we've talked about how the heart pumps blood around the lungs and the body, this dual circuit, and of course the timing and the balance between these circuits is exquisitely managed, isn't it?
It's fantastic. And it's one of the reasons, of course, it's very difficult to build the artificial heart, to get that degree of precise engineering. What we have got is partial artificial hearts, where instead of taking the whole heart away, you leave it there and you just help the blood. Some of the blood is taken out through a tube at the bottom of the heart and put into the aorta with a little impeller pump in between. And so it's an assist device. And those have been much more successful, a left ventricular assist device. And so there's a lot of people who've had those in place for some years. Theoretically, they're waiting for a transplant, but actually or really there's so few compared to the need that they often end up on the support device for quite a long time, years.
And these LVADs, I mean, this is the best option now for people waiting for a heart transplant.
For severe end-stage cases, yes, that will certainly give them a much better quality of life. Although the fact is that they do have physical limitations. Actually, what you've got is an external battery, and people carry them on their shoulders, and you've got drive lines coming through to the battery, and so you've got something that breaches the skin. So that's a challenge to keep clean and not have infection coming in through that. And then you obviously have to have your battery charged up. And the first, one of the first people to have this, he was walking down, I think it was down Oxford Street, I may be wrong. It had a bag snatch. It was snatched as if it was a bag. Luckily, he had enough charge to get back to a battery. But, it's a bit worrying.
My God, yeah.
Yeah.
That's something you definitely don't want stolen.
No.
But the incredible thing about the LVADs is that people don't have pulses, right?
Yes, it's always the joke to make the young intern come and try and find the pulse.
And the reason there is no pulse is because it's not trying to recreate the beating heart. It's going for a more smooth flow of blood in the body, and that then removes the pulse.
That's right, because the first ones try to be pulsatile like the heart and compress the blood like the heart does. But because they were metal plates, they caused some damage.
And so we talked about the cardiomyocyte, the heart muscle cell. And could you just describe for me the moment that you first saw these under the microscope?
Yes, they are quite tricky to get out of the heart because each cell is connected to every other cell about 7 or 8 ways. There's lots of junctions between the cells. It is a very tricky thing. And so when we first saw these cells, we were just we were so happy. And when we put the electrical current across them and saw them beating away, just like little hearts. That was just amazing. In fact, we were so bad in the beginning. We had a little motto in the lab called "Una sed bona," which was one cell but a good one. So we were happy if on a given day we had one cell to experiment on, but as long as it was a good one. Yeah.
And cardiomyocytes have a hard time being alone. So, unlike other types of cells, like skin cells, for example, when you try and grow them outside of the body, they really do need company or die.
That's right. Their structure, which is this beautiful structure, so it's sort of very stripy. So you've got the overlapping of the muscle fibers produces a kind of stripe across them, and you can see that beautifully as they contract. That gradually disappears with time over a couple of days. So you can probably they're good for a day. They're not bad for 2 days, but after that, they just become amorphous masses. And similarly, when we have, and we're going to talk about this later, when we made some very young cells from stem cells, they start off quite amorphous, and you have to give them mechanical forces to give them the idea about how they should join up and how all the fibers should align together. That's why we kind of exercise them.
Yeah. So, heart muscle cells need to work out like we do.
Yeah, they do. Yeah, they need to be together and they need to work out. Yeah.
There's a part in your book where you talk about discovering these hidden hills and valleys on the surface of these heart muscle cells.
One of the good things about having an individual heart muscle cell out of the heart is that you can look at it in very great detail. But you're always sort of limited by the wavelength of light. And so if you want to get down to the really deep structures, you have to get below that. People in our place, Yuri Korchev and Julia Gorelek, they designed, and classically, Yuri made the first one out of something he found in a skip, one microscope he found in a skip, and he designed this. And it's basically that you have a very sharp, narrow glass pipette which scans across the surface of the cell. And the trick is to keep it at exactly the same distance from the surface. The cell is electrically charged, and so it can sense the electrical charge on the cell. The surface is very indented, it's very regular hills and valleys. It goes over those and it shows you a contour map of those, and particularly the deep tubules that go into the cell, and they carry the electrical charge right down into the cell to do its work in releasing the calcium.
Incredible. All because these hills and valleys are smaller than the wavelength of light. And
That's
so
right.
you
Yes.
need to have another way of seeing them.
Yeah.
So we mentioned that the heart breaks down. Now would be a good opportunity to just define heart failure and heart attacks, which are kind of two different components of heart disease.
So a heart attack, which I expect people are familiar with from all the television programs, it's usually caused by a blocked blood vessel. So your heart is hurting because you've got build up of lactic acid in it. So you're feeling that you could feel nauseous. Your pain might be down your arm, might be felt in your back or your jaw. You feel slightly faint. You'll go grey. You have a feeling of doom. I mean, they say you have a feeling that something terrible is happening, because something terrible is happening, actually. So fair enough. But it's a feeling that's much more than you might expect from just the pain. So what's happening there is, so I say the vessel is blocked. You've got this lactic acid building up, and the muscle is not getting oxygen. And so it starts to die. And so that's why you must move really, really quickly, because time is muscle. You've got to get to the hospital. What will probably happen is that you'll have a catheter put down your vessels, which through your arm or through your leg up into the heart. It will remove the blockage and usually put in a little narrow metal tube to keep the vessel open, a stent, which become more and more sophisticated these days. So that's the heart attack. What happens after that depends on how much muscle has died, how quickly you've been treated. And so many people are fine. About 7 out of 10 people will recover. If there's a lot of muscle that's died, after a while you can go into something called heart failure. And this is where your heart is really not pumping blood enough. And symptoms of that are different. Fatigue again, but you have water retention. You have swelling of your legs, for example. You might be breathless because you've got water collecting around your lungs, but it's just a sort of very gradual thing. And so about half of heart failure is due to something, somebody having a heart attack. There are other things that cause heart failure, and the diabetes and the cardiomyopathy is one of them, high blood pressure, chemotherapy. So there are a number of other things, and they feed into this heart failure. And actually, this is where I was mentioning the sort of survival mechanisms. What's happening is your body is sensing that your heart isn't performing adequately and trying to stimulate it. And this produces more damage. It's the things that your body is doing. They're either making your heart beat harder with adrenaline, or they might be thinking you've got an injury and trying to retain water to give your heart more to work on. Both of those, when you have them for not an emergency, but for a long, long time, cause further damage. And so a lot of the drugs you use are actually just to stop your body damaging your heart further, actually, when you've got heart failure.
It's, as we discussed earlier, it's another example again of evolution harming more than helping. Because no doubt, if you see a line behind the bush, having adrenaline helps get the muscles filled with blood. And so you're good to sprint off. But in this context, it actually causes long-term damage to the heart. And you mentioned how chemotherapy can damage the heart, and that's very relevant at the Champalimaud Foundation, because we don't just have labs here that do research in cancer. We also have a clinical center as well that treats cancer patients. And so what oncologists found was that successful treatment of cancers often had the effect of causing heart damage in the patients. And this was first seen, I think, in breast cancer patients.
The reason really for the breast cancer was because it was very successful therapy.
Yeah.
So people were now recovering and living quite a long time. And so those effects were starting to emerge because they had a large population that they could study. And what you're doing with the cancer drugs is killing off fast growing stem cells and cells like that. Now, the heart really doesn't have very much in the way of regeneration. There was many, many very vicious arguments for a very long time about whether it had any at all. And it has a little bit of regeneration, and that's just about probably enough. So 1% per year dropping down to about 0.5% per year as you get older, of your cardiomyocytes will be regenerated. And so actually, if you do the math, if you live to about 80 or 90, half the cells in your heart will have been there your whole life. And so pretty good for a little cell like that, and which is just incredible. So when you've got something that kills off that little bit of regeneration, then you can see that that's a problem.
And actually, this is something that the heart shares with the brain, because the brain also has few dividing cells. And for a long time, people thought that there weren't any new brain cells being made. And often we talk about feelings and emotions coming from the heart. And it turns out that the heart creates emotions as well as responds to them.
Absolutely. The heart has a little mini brain within it, a sort of distributed network of ganglion clusters of cells. And so when we talked about that fight and flight response, that's an emergency response. And so the body wants to do it without bothering the brain first, because if you had your brain was thinking, shall I run away or shall I not run away? No, you don't have time for that. So there's a reflex from your sympathetic nerves, which are autonomic nerves, which control all those things that are underneath your voluntary control, so like breathing and digestion and things like that. And so the speeding up and slowing down of your heart is in that system, that one you don't really control. So there are sensory nerves in the heart which then tell the brain, look, I've speeded up, something's going on here, just have a look round. And if you play somebody a speeding heart and you tell them it's their heart, the same thing will happen. They will get anxious. They will look around. Oh no, what's going on? And so the brain will then, if it sees the threat, will double down on that, and you'll carry on running. If it doesn't, then you should just deactivate in a way. But of course, there are lots of things that make us anxious and make your heart race that now your brain can say, I don't really know where this threat is coming from. And so you get this kind of free-floating anxiety, or anxiety attached to things it shouldn't be attached to. It's why one of the first things you have to do if you're an anxious person, you're given exercises like breathing. What it does, it activates the vagus nerve, which is the opposite of the sympathetic nerve, which calms everything down. And so you're using your breath to activate the calming system that will take you back down when the sympathetic nerve has been activating you.
Since we're talking about the autonomic nervous system, there's a lab at the Champalimaud Foundation that recently discovered that fruit flies' hearts respond to danger much like our own. And speeding up if a fly runs away and slowing down if it freezes. And this finding was unexpected because flies, of course, lack an autonomic nervous system, which is the system that typically manages these responses in us vertebrates.
That's very interesting.
And there's also a fascinating study where you talk about researchers flashing frightened faces at different points in a heartbeat.
Yes, so one of the things, we're very social people, obviously, and one of the things that makes us frightened is other people being frightened. And so that social contagion, that's a really important thing for getting out of danger. And the way you understand that a face is frightened is by, I'm trying to do it now. If you open your eyes really wide, you can see the white all the way around it.
Yep.
Okay, good. I'm relieved that worked. So that, to see the white around your eye, makes you understand that somebody's frightened. So the experimenters, Sarah Garfinkel, she did a flash of either somebody with just a neutral expression or somebody where you could see the fright subliminally. So it's really a very quick flash, and flashed either at the beginning of the heartbeat, so when your heart's in systole, or when it's relaxed afterwards. And there's a bit of a difference in your blood pressure between those. So there's a little bit more activation when your heart is at the peak of beating, and less so when it's relaxed. And she then measured fear responses from skin, a slick of sweat on your skin resistance goes down. And so if you flashed the face at the end when the heart was relaxed, you had a small fear response. But if you flashed it when the heart was activated, you had a high fear response. That's how subtle it is. Within the space of one heartbeat, you can tell the difference in the sensitivity of that system to activate things. And so at the top, you're just coming to the peak of your sympathetic activation. And so you've got more adrenaline, if you see what I mean. And so you're already activated, and this frightened face gives you even more fear because you've amplified the effect.
So these baroreceptors, these sensors that detect the changes in blood pressure during the heartbeat cycle, they send a message to the brain.
That's right. It's sort of it's the autonomic nervous system. Yeah, right. That's right. Exactly. You've got more activation of the heart than the brain. Yeah. It's all without you being frightened or knowing you were frightened.
Right.
Because it's subliminal. You're seeing a very, very rapid thing. So it shows you how rapid that message to your brain to get going is, and how it's below the level of your consciousness.
And as you say, this can happen incredibly quickly. So as you write, if someone opens a phone conversation by saying, I have some bad news for you, I can guarantee your heart rate will be soaring by the word 4.
My mother always used to, whenever she was telling us anything, she would always open with, it's all fine now, and then tell me the bad thing. She understood this.
Smart.
Yes.
And so this brings us to our next topic, because we talked about how the heart can create emotions, but of course emotions have an effect on the heart too. One example, broken heart syndrome.
So people who have broken heart syndrome, we call it that because they've had an emotional shock.
Right.
And so that's why it's called it. And it's usually or often bereavement. You're statistically more likely to die just after your spouse has died, as the risk declines over about 6 months to a year. And so those kind of things, there's a really huge adrenaline release, and that's definitely attached to emotion. So that's broken heart syndrome. It can happen in men and women, but it's more often in women and more often in postmenopausal women, sort of the transition as you're losing your estrogen hormones.
Right. So this is the example that I mentioned at the outset when I read the passage from your book in Japan, for example, after the earthquake.
That's right, yes. And so natural disasters is where it has often been seen. They were taken into hospital, but these people who came in thinking they were having a heart attack, but actually they often got better. And so they really weren't sure what was happening. And they had this different imaging system, so you could see the heart in silhouette. And they could see that, usually heart's going like that, but the top, where the blood comes out, was really, really contracting very hard. But the bottom, the apex, was like still almost, and it would look like on the picture, it looked like an octopus pot because you had the tight constriction here and then the ballooning there. And apparently that's the shape of a pot if you want to catch octopuses. So that's another useful takeaway tip from this podcast.
Well, we have a lot of catching octopuses in Portugal because the Portuguese do like a good octopus, or polvo as they call it.
Oh, right. So even more better for our Portuguese listeners. Excellent. And now I think that lots of hospitals do understand that this is a possibility, and they do this imaging, and we have got much better imaging now, and they will see this. And this is where what the adrenaline has done is to flip into a different mode where it's frozen part of the heart. And actually, we did a lot of investigations on what this is. But if you try to stop it doing that, what happens is it then goes into ventricular fibrillation. So it looks like that what's happening there has some kind of benefit in a way, that it's stopping the arrhythmias. And it has some parallels with playing dead. Animals play dead deer, the baby deer, when they're alone, if the mother's gone off to get some food. If they hear a predator coming, they're able to really slow their heartbeat right down. There is a reflex that does this, that really slows the heartbeat right down. And so the predator can't hear their heartbeat.
And intense experiences, exercise can trigger ventricular fibrillation. And so people may be familiar with stories of footballers in the middle of a match suffering from sudden cardiac arrest. And this is when the ventricle no longer contracts in a coordinated way, and different areas are now contracting and relaxing at different times and speeds, and kind of wriggles and churns without ejecting any blood. And I think surgeons who see this during an operation when the chest is open describe it as the bag of worms appearance. Could you just give us an insight into what is actually going on when a footballer does suffer from this sudden attack?
So what you're looking at then is an overdose of adrenaline, pretty much. They're exercising, it's very extreme exercise. And what's happening is that the cells, individual cardiomyocytes, are being stimulated to increase their calcium coming in and out. But actually, it's such a strong stimulation that the different cells start to go overboard and have little arrhythmic contractions themselves, so that the one is contracting at a different time to the other. So it's an adrenaline overdose that's doing that. And it depends a bit on what your heart is like as well. People can be more prone to it. And one of the things that makes you prone to it is having a thick heart. And often athletes will develop this hypertrophy. And if you have a mutation or something, you can get a pathological amount of thickening of your heart. And so that when you get the adrenaline, because it's very thick, it is more difficult to be coordinated. And so actually a lot of football teams now have a heart checks, particularly as this happens at puberty. So these young players who are going through puberty, they might have been perfectly fine before, nothing at all, but they might just have this hypertrophy that would predispose them to the adrenaline effect.
And so you talk about how male and female patterns of heart failure can differ.
It's very clear, honestly, that young women are less likely to have a heart attack than young men. So there is a cardiac benefit for estrogen, especially when you're premenopausal. Obviously, then you start to lose it. I have to be a little bit careful here because this is a relative thing. So probably men are 2 or 3 times more likely in their younger years to have this. But that doesn't mean that women don't have it. And there's been a lot of problems because people have understood this one fact about it's a male disease, that women have been very much overlooked when they've gone into hospital for heart attacks. And so we've been trying to redress the balance here and say women can have heart attacks, they shouldn't be ignored.
But the treatment that they receive often differs, and women with heart disease are less likely to receive optimal treatment. And you also mentioned that heart attack survival rates are 2 to 3 times higher for female patients treated by female physicians.
And that was a particular study. It's enormously helpful that we have these really big studies going along. So this was 1.3 million heart attacks in Florida. And exactly as you say, in that study, I'm not saying it happens everywhere, but the ones who were treated by female physicians were twice as likely to survive, more than twice as likely to survive, which obviously is extremely important. There was things like as the male doctors gained experience with women, that got better, statistically speaking. If they had been working in a balanced team with male and female colleagues, then they also were better. The one problem being that in many places, particularly in the US and the UK, cardiology is a very male-dominated profession still. Certainly, the British Heart Foundation have been working very hard to try and to support those women who are, particularly the interventional cardiologists, who are the ones that put the stent in for you. I think it was about 7% in the population that we were looking at in terms of the number of females in that group.
And one hypothesis you put forward for why estrogen may have a protective effect against adrenaline is pregnancy and childbirth. Evolution, there's a strong evolutionary pressure to protect the mother at that moment. And so maybe estrogen has some kind of protective effect for that very reason.
Something we've never, unfortunately, been able to test experimentally. My colleague, who done much of the work in this area, Alex Lyon, did try to persuade his wife, he has 3 children, to be wired up during labour and have some blood tests, but I think she felt she'd done enough by having 3 of her children, so it's fine.
All All right. So now let's come to the final chapter then of this podcast and indeed your book, Healing the Heart. Now, one interesting thing I saw was that babies actually can recover much more easily from massive cardiac damage than adults. And so people have actually been looking now at stem cells and trying to use what are called pluripotent stem cells.
They're stem cells. They've really got potential. They're pluripotent. They can't make the whole baby, which is the totipotent cells, which is the fertilized egg, but they can make all the cells that would go into the baby. But for a human, of course, if you're getting those, you mean you've dissected out a fertilized egg, and people have consented if they've had fertilized eggs for that to be done. But there's a lot of ethical problems. That could have become a baby. But somebody won the Nobel Prize for this year, Shinya Yamanaka, because he managed to work out what factors that would take a normal cell, like a skin cell, any skin cell from you, and push it back to become a stem cell, take it, sort of strip all the messages that make it a particular kind of cell, and get it back to its earliest form.
To turn back the clock.
So turn back the clock, exactly, and make them like they were in the womb or the fertilized egg. And these are called induced pluripotent stem cells because you've made them. And those can do exactly the same thing. You can make them into all the cells in the body. And so using those particularly, people have spent a lot of time and energy getting the conditions exactly right to produce cardiac cells. So now we're actually very good at that. We can produce large numbers. I was making strips of cells about as big as an elastoplast, about as big as my thumb, which were beating away. I mean, you can make them as big as you like. People have made them about the size of a hand, so they'd be right to put over a human heart. It's just how much money you've got, really.
So here we basically have a skin cell, for example, which is turned back into a stem cell that is then treated with various factors to turn it into a heart muscle cell. And then you can grow that in the lab and indeed strengthen the muscle, giving it essentially a workout over weeks and months so that you have a heart muscle which is ready to be implanted into someone with a failing heart.
Yes, that's right. What we've got now is a sort of engineering problem. First, are you going to use your own cells to do this, or are you going to use somebody else's? If you use your own cells, then it takes a while. Theoretically, it's matched to your heart. Okay, so theoretically, if you were to put it in, you wouldn't reject it because it's you. But then you have to do that for every single patient. So that's the thing. Economically, will that work?
Your subheader for this chapter was "Your heart is in the mail."
Yes. Yes. The other thing is you have a donor, and so you have somebody who donated it. You don't have to wait 6 months. You can make it into the shape you want. You can implant that. But then you have to give the person immunosuppression. And if you'd give them a heart transplant, they would have had to have immunosuppression. So what's the difference? Well, the difference is immunosuppression is nasty, and you have to be really sure this thing is going to work before you can justify giving a patient immunosuppression. Then the third thing is that the product itself, the tissue itself, is this young tissue, so it does have this pacemaker activity. So one of the problems we've been having is that, and say you do this for a pig heart because it's large like a human heart, or inject it in, or you put it on as a patch, eventually it will integrate, and it does have good benefit for people who have scars from a heart attack. But in the meantime, because these are young cells, you have to disrupt the structure of the heart to get it in. So that disrupting of the structure is dangerous. They have their own pacemaker, which is dangerous because you don't want another pacemaker in there. You've already got a pacemaker. And what's happened is that you get a period where you get very violent rhythm disturbances from putting these new cells in. But this is again a barrier to having this as a therapy. And when you get to people, the firms, which have to pay out large amounts of money, and they have to choose between a drug, which they can keep giving you for the rest of your life, or something that's your cells, so they can't really charge you for those, you can see that the economic incentives are very difficult.
Yes, the economic incentives for pharma to take this approach are not really there. And so, this idea then of growing a heart in the lab from either the patient's stem cells or someone else's, then strapping it back in to the failing heart to try to restore it, does have these drawbacks that we've discussed. And then also the fact that there may be, some stem cells which haven't differentiated yet, and that potentially could turn into a cancer.
Mm-hmm, mm-hmm. Yeah, so if you've been able to make the product early from somebody else's cells, you can test for those kind of cells. When you're doing it from your own cells, you don't quite have the quality control in the same way. Still some evidence coming out about exactly how many undifferentiated cells would be a danger. The latest evidence that I'd seen is even a few would be quite a danger, actually.
Right. But of course, even if this approach doesn't work, this idea of kind of strapping the heart muscle into a failing heart to try to bring it back to its normal function, it's still incredibly useful for what people call a disease in a dish approach. And this kind of chimes in with personalized medicine, this idea of taking approaches, individualized approaches to the patient in question. So you can take their skin cells, which then become heart cells, which you can then test drugs on and see how effective the drugs are, and potentially which drug is best at restoring the function of the heart muscle cells. Is that correct?
That's right, that's exactly right. And this has been the great success of these. They're great for understanding the relationship between a mutation or variant and the outcome because you can genetically engineer them quite easily. So you can knock out the gene and see if that cures it. You can knock in the gene. And so it's extremely precise and informative to understand how you get from the mutation to the disease. Also, then to test out old drugs and see if any of them are good to be repurposed for a disease. And there's been some good successes along the way.
Yeah, actually, it reminds me of Rita Fuel's lab at the Champalimaud Foundation, which is at the moment leading a clinical trial that uses zebrafish embryos
Mm-hmm.
as avatars for cancer patients. And in this approach, doctors collect tumor cells from a patient and implant them into transparent zebrafish embryos. And by testing different drugs on these embryos, they can quickly see which treatments seem most effective for that patient's tumor. And so, if the avatars prove to be accurate, they could help doctors choose better therapies faster and spare patients from toxic, ineffective treatments.
Yes, absolutely. That's a fascinating example, and zebrafish are a wonderful model. The other thing, as you say, sparing people from toxic things, even drugs that don't work on the heart, like the cancer drugs, they're to do with cancer, they have an effect on the heart. And so many other drugs for other organs have an effect on the heart. And so you can choose, if you've got a large number of these, you can choose the ones that do or don't have cardiac toxicity. The cardiac toxicity is one of the biggest reasons that drugs fail as they go through the system.
So maybe we can end then, Sean, with you just giving us a taste of this gene therapy journey that you went on.
We've been looking at the cardiomyocytes. We've been looking at the cardiomyocytes from hearts of people who have heart failure. We saw that there was one protein that handles the intake and output of calcium and putting it into intracellular stores. It seemed to be particularly changed in heart failure. And so we used what was a new technology at the time, a way of infecting the cells with a cold virus, an adenovirus, and carrying in the gene for this thing called SERCA. And we could see that when we had the cells from patients with heart failure, when we put that gene back in again, they behaved like normal cells. They behaved like fresh hearts. So we could see that that was a great target.
Right. And just to be
Um,
clear, this is different from stem cells now. Here we're talking about delivering a specific gene using these viruses into the patient's cells in order for their heart cells to then express this protein.
that's right. We chose a virus which was the best around at the time, which wasn't quite so problematic at producing an immune response. Because obviously, you put a virus into a person, they respond to it and they produce antibodies. Because it was, you were one of the first people doing it, but people didn't know what was going to happen, how dangerous it might be. It was about 7 years to do the paperwork. We had to decide all sorts of things, whether we treated the patient in the hospital and we kept them apart, barrier nursing, from everybody else until the virus had cleared their system. So when we gave them the virus, they didn't give it to anybody else. The virus didn't replicate. We stopped it replicating, so they shouldn't catch it. You wouldn't be able to catch it. But nevertheless, if you had it in bodily fluids, then people who are nursing you or your relatives could be affected by it. And of course, we didn't know how infectious it was going to be. And I can tell you that there are many more bodily fluids than you would imagine, to be honest. Very, very troublesome bodily fluids, very troublesome. And this seemed to be just logistically impossible. You would carry on finding the virus and or bits of the virus, even in sperm for months and months, years. So really very difficult. Then the site that we were doing these tests or treatments of the patients now became a site of release of genetically modified organisms. So we had to go through all the paperwork you had to do for like genetically modified wheat or something like that, because it was a potential release of a genetically modified organism. We had to go out to public consultation. It was just crazy times.
Yeah, no, I mean, the regulation, I mean, it sounds like the regulatory hurdles were pretty high. So the infection of patients with this virus and their journey home from the hospital
and
could
when
potentially
they see
be
their
classed. Sorry?
Yes, exactly. Their journey home from the hospital. Yes, that's classed as release of a genetically modified organism now.
Right.
Anyway, it was 100 times more complicated than that. I mean, we could be all here all day if
Sure.
And it would be good therapy for me, but perhaps not for you. And the bottom line was it didn't work.
Because the cells didn't take up the virus.
Yes,
they didn't take up
the heart was just so resistant to virus, much more resistant. And the heart itself was the one organ that didn't really want to take it up at all. And there's a big beat challenge by the British Heart Foundation. An international group has got together to use the newer kinds of gene editing like CRISPR, which are much more sophisticated than viruses and don't have the problems of viruses.
Well, and let's see what future experiments hold. So I think we've come to the end of this chat. It's been a remarkable tour of the heart, and I hope our listeners now know a few things about the heart they didn't before. Could you mention any useful resources for listeners?
So the British Heart Foundation do have a website, so that's very, very useful in terms of the basic symptoms of heart disease. It has a lot about the treatments and what they mean and what they mean for you. It has a very good section on women and heart disease, particularly. They did a report on women and heart disease. And you'll see they're very interesting new research, like, as I was saying, the Big Beat Challenge, try to actually change the genetic makeup of the heart.
There's also an organization called Women Heart, which strives to improve the lives of women with or at risk for heart disease. So we can include links to BHF and Women Heart in the show notes as well. How can people get in touch with you, Sian?
I think you'll probably be able to find me on the Imperial website. If you just type in Sian Harding Imperial, you can get in touch with me.
Super. And lastly, any book, fiction or non-fiction, that you would like to recommend to our listeners?
I really liked one that came out recently called The Immune Mind, which had a very kind of similar feeling to what I've been saying, in that first, that the brain is more affected by the immune system than you think, but also the immune system causes behaviors, sickness behaviors, and there's a big link between those and some mental health conditions because it changes the way you think. And the basic underlying theme that these things are important for you to survive. You're being threatened by these bugs. You've got to survive them. You've got these mechanisms that are finely tuned to help you survive them. But they can get out of hand and they can cause problems. Evolution doesn't care if you are happy or unhappy or whatever, anxious or whatever. Doesn't care about that at all. You know, they just want you to survive. And so that's the thing. That's what it's geared up for, survival.
And who was the author of the book?
Monty Lyman.
Monty Lyman.
Lyman.
Okay, great. All right. Well, a heartfelt thank you, Sian.
Thanks very much.
That was really wonderful.
It's really great to talk to you, and great work in your foundation as well.
Thank you. Thank you.
Okay,
All right. Well, it's bye from me.
that's bye from me.